Hello there. Welcome back to Midnight Ocean. I'm Velen, your guide to traveling through the wonders of the ocean. Tonight, we're going to explore some of the most stunning marine ecosystems of the oceans. If these cozy moments calm you, I invite you to subscribe, share a comment, or leave a like. Now, get comfortable, take a deep breath. Don't be afraid, you're safe here. Let's dive deep into the sea and let our imagination guide us to a peaceful and deep night. Australia's Great Barrier Reef, stretching 2,300 kilometers and home to 1,500 fish species, experienced in 2024 the most spatially extensive bleaching event ever recorded, losing between 14% and 30% of coral cover across all its regions in just 1 year. Puerto Rico's bioluminescent bays concentrate up to 700,000 dinoflagellates per gallon of water, turning every movement on the surface into a trail of electric blue light. The abyssal zone, between 4,000 and 6,000 meters deep, harbors organisms that have never seen sunlight and accounts for 60% of the ocean's total volume. The subglacial polar ocean, beneath Antarctic and Arctic ice, supports an entire food chain that starts with algae living on the underside of the ice and ends with whales. Hydrothermal vents along mid-ocean ridges sustain ecosystems based entirely on chemosynthesis, with zero dependence on solar energy. The Sargasso Sea is the only sea on the planet with no coastline, bounded by currents, not land, and the only place where both European and American eels go to die after spending decades in rivers. Blue holes are submerged vertical caves with anoxic zones that preserve fossils from ice ages. California's kelp forest is the only ecosystem where sea otters float in the canopy, while leopard sharks patrol the bottom. The Coral Triangle holds 76% of all coral species on the planet within just 1.6% of the ocean's surface area. And the Hadal Zone, below 6,000 meters, contains plastic, amphipods, and bacteria that have existed for millions of years without any contact with the surface. Think of the ocean not as a surface, but as a vertical tower. The sunlit layer, the first 200 meters, is where nearly all marine photosynthesis takes place. Below that, light gradually fades until it disappears completely. This layering isn't just optical, it defines who eats whom, who survives, and who never makes it up. Each depth band has its own temperature, pressure, and chemistry, shaping organisms that are radically different from one another, as different as a parrot and an abyssal worm. Understanding this vertical gradient is the starting point for grasping why the ocean, being a single continuous body of water, harbors ecosystems as irreconcilable as the ones listed above. The first layer, the epipelagic, from 0 to 200 meters, is the most familiar and the most studied. Phytoplankton, a collection of photosynthesizing microorganisms, produces more than 50% of all the atmospheric oxygen on Earth right here. These organisms are invisible individually, but together they color the sea and form the foundation of virtually all marine life. Zooplankton feed on them. Small fish feed on the zooplankton. Larger predators close the chain. Without this thin sunlit layer, the entire ocean would collapse nutritionally. This is where coral reefs, kelp forests, and bioluminescent bays exist, all within a tiny fraction of the ocean's total depth. Below 200 meters begins the mesopelagic zone, informally known as the twilight zone. Sunlight arrives only as a faint glimmer, not enough for photosynthesis. Creatures in this band have developed extreme adaptations. Huge eyes that catch any stray photon, transparent bodies that make them harder to spot by predators, and their own bioluminescence used for communication and hunting. The daily vertical migration is a defining phenomenon here. Organisms rise to the surface at night to feed and return to the depths at dawn, carrying carbon downward in the process. This migration is one of the largest movements of biomass on the planet, invisible from the surface. The bathypelagic zone stretches from 1,000 to 4,000 meters. No light, no photosynthesis, no plants. The pressure is equivalent to hundreds of atmospheres, enough to compress human lungs down to the size of oranges. Organisms in this region feed on marine snow, organic particles that continuously sink from the surface, a slow rain of fragments from dead organisms, feces, and mucus. This snow is the link between worlds, connecting the productivity of the sunlit surface with the perpetually dark depths. Fish like the viperfish and the vampire squid inhabit this darkness, equipped with bioluminescence and oversized jaws to capture whatever protein they can find. The abyssopelagic zone, 4,000 to 6,000 meters, covers more than half of the ocean's total volume. Here, the temperature permanently hovers between 0 and 2 degrees Celsius. Pressures exceed 600 atmospheres. With no light and no significant thermal convection, the environment is stable to the point of geological monotony. Yet the sandy bottom, known as abyssal mud, harbors a surprising diversity of invertebrates: sea cucumbers, polychaetes, bivalves. Small, slow, metabolically frugal organisms, waiting for organic particles that sink from kilometers above. The biodiversity here is invisible to the naked eye but real, spread across expanses larger than any land-based forest. Hydrothermal vents break up this nutritional desert with oases of intense chemical activity. Located along ocean ridges, underwater mountain ranges where tectonic plates pull apart, these vents expel superheated water rich in hydrogen sulfides, methane, and dissolved minerals. Chemosynthetic bacteria oxidize these inorganic compounds to generate energy, completely replacing the role of the sun. Giant tube worms, yeti crabs, blind shrimp, and squid colonize these hot spots at densities impossible anywhere else in the abyss. Discovered in 1977, hydrothermal vents prove that life does not, fundamentally, need sunlight. The Sargasso Sea holds a unique position among all the seas on Earth. Located in the North Atlantic, it doesn't touch any continent. Its boundaries are 4 ocean currents that rotate around a center of high atmospheric pressure. Floating algae of the genus Sargassum cover its surface in dense mats, creating 3-dimensional habitat for juvenile sea turtles, octopuses, seahorses, and crustaceans. European and American eels travel thousands of kilometers from separate river systems to spawn and die here, a migratory behavior whose navigation mechanism science has yet to fully decode. Blue holes are geological structures formed during glacial periods, when sea levels were much lower and limestone caves developed on dry land. As the ice melted and sea levels rose, these caves were flooded, trapping inside them layers of water with no circulation, anoxic zones where oxygen is virtually absent. Under these conditions, conventional bacterial decomposition doesn't occur, and organic matter deposited there is preserved indefinitely. Belize's Great Blue Hole, 300 meters wide and 124 meters deep, is the most famous, a circular window into the geology of past eras, filled with stalactites formed when it was still a dry cave. California's kelp forest represents a 3-dimensional ecosystem of rare complexity. Giant kelp, which can grow up to 30 centimeters per day under the right conditions, forms an underwater canopy analogous to a tropical rainforest on land, with distinct layers of light and shadow. Southern sea otters live in the surface canopy, wrapping themselves in the kelp to sleep without sinking. Leopard sharks, bright orange Garibaldi fish, and rays sweep the bottom. Each layer has its own distinct fauna. The otters, by feeding on sea urchins, keep those animals from destroying the base of the kelp, a predator-prey relationship that holds the entire ecosystem together. The Coral Triangle, spanning the waters of Indonesia, Malaysia, Papua New Guinea, the Philippines, the Solomon Islands, and Timor-Leste, contains the greatest concentration of known marine biodiversity on Earth, covering just 1.6% of the planet's ocean area. The region is home to 76% of all known coral species in the world, as well as habitat for 52% of Indo-Pacific reef fish and 6 of the 7 existing species of sea turtles. The Hadal Zone, below 6,000 meters, rounds out this inventory as the most extreme environment of all. Pressures that would crush military submarines, total absence of light, temperatures near freezing, and yet, crustacean amphipods, adapted bacteria, and as recently documented, fragments of human plastic that reached the deepest trenches before any researcher ever visited them. The Great Barrier Reef is 2,300 kilometers long, visible from space, and the largest living structure on the planet. The 2024 mass bleaching event had the greatest spatial reach of any bleaching event ever recorded on the GBR, with high to extreme bleaching observed across all 3 regions, North, Central, and South. In 2025, 48% of surveyed reefs saw a decline in coral cover, 42% showed no net change, and only 10% showed an increase. The numbers by region: hard coral cover in the northern GBR dropped from 39.8% in 2024 to 30%, the largest annual decline ever recorded for that region. In the south, it fell from 38.9% to 26.9%, also the largest annual decline ever recorded for that region. The Great Barrier Reef has experienced mass bleaching events in 1998, 2002, 2016, 2017, 2020, 2022, and 2024, 6 of them since 2016. The average gap between mass bleaching events on the GBR has been cut in half since 1980. Coral is not a colorful rock. It's an animal, technically a polyp, that lives in colonies on top of a calcium carbonate skeleton. It builds itself over the course of decades. Inside its tissues live symbiotic microalgae called zooxanthellae, which photosynthesize and supply the coral with up to 90% of its energy. This relationship is the reason coral reefs exist only in shallow, clear waters. The algae need light. When water temperature rises beyond a narrow threshold, usually just 1 degree Celsius above the historical summer average, the coral expels the zooxanthellae. Without them, the transparent calcium carbonate skeleton becomes visible. The coral turns white. That's bleaching, not immediate death, but a critical state of starvation. Bleaching doesn't kill coral instantly. A bleached coral is still alive, but without its photosynthetic partner, it's malnourished and vulnerable. If temperatures drop within weeks, the algae can recolonize the tissues and the coral recovers. If the heat lingers for months, death becomes inevitable. Cells without energy can't keep living structures going. The 2024 event was particularly destructive because it combined high temperatures with prolonged duration. At One Tree Reef, scientists monitoring 462 individual colonies found 80% bleached by April 2024, and by July, 44% of the bleached colonies had died. Fast-growing corals, which had driven recovery after previous events, suffered 95% mortality. The Great Barrier Reef is not a single uniform structure. It's 2,500 individual reefs spread across 346,000 square kilometers. Each reef has its own distinct history of exposure to temperature, cyclones, sediment, and human pressure. In 2024, all 3 sectors, North, Central, and South, were simultaneously hit by the same event, something without precedent in the historical record. The Great Barrier Reef lost between 1/4 and 1/3 of its hard coral cover across the 3 main regions, according to the Australian Institute of Marine Science. In some areas, the loss reached 70%. That's the equivalent of losing between 25% and 33% of a continental rainforest in under a year. What makes 2024 stand out isn't just the scale, but the historical context. Previous mass bleaching events on the Great Barrier Reef occurred in 1998, 2002, 2016, 2017, 2020, and 2022. Before those years, there's no evidence of events that widespread in 500 years of coral records from the Great Barrier Reef. The acceleration is alarming. 2 events in prior decades, 6 events in the following 8 years. The average gap between mass bleaching events on the Great Barrier Reef has been cut in half since 1980. Corals that once had decades to recover now have months, not enough time to restore structures that take years to grow. The 2024 bleaching didn't act alone. The declines are mainly attributed to coral mortality stemming from the large-scale 2024 bleaching, compounded by the combined impact of 2 cyclones in December 2023 and January 2024, freshwater flooding, and outbreaks of crown-of-thorns starfish. This starfish, Acanthostera plancey, is a voracious coral predator that explodes in numbers when ecological imbalances wipe out its population controllers. Thermal stress plus cyclones plus predation plus flood sedimentation created a convergence of factors that no reef could easily withstand even if facing just one of them. The 1998 bleaching was the first recorded with adequate surveying coverage. Caused by the most intense El Niño of the 20th century up to that point, it wiped out large swaths of coral worldwide and put scientists on alert about reef vulnerability to warming. On the Great Barrier Reef, the 1998 event primarily hit the north. The second event, in 2002, reached a similar scale. Then came 14 years without a major event, a gap that seemed natural at the time, but was really the product of favorable ocean conditions. In 2016, an El Niño of comparable magnitude triggered severe bleaching in the northern sector, killing 50% of shallow water corals in specific areas. In 2017, before any recovery, a second event struck the center. In 2025, 48% of surveyed reefs saw coral cover decline, 42% showed no net change, and only 10% showed an increase, with stable or growing reefs concentrated mainly in the central region. This pattern reveals a hierarchy of damage. The north and south, the 2 ends of the Great Barrier Reef, have progressively lost the ability to absorb new events before recovering from previous ones. The central region, topographically sheltered and geographically positioned between the hottest zones, held up better but was not immune. Fast-growing corals, mainly those of the genus Acropora, whose branching, tree-like structures build the 3-dimensional framework that gives the reef its structural complexity, are simultaneously the fastest to grow and the easiest to kill. These corals were extremely vulnerable, and their loss had already been flagged as a serious concern. They were precisely the organisms that had helped drive partial reef regeneration after previous events. Losing the fastest rebuilders means any future recovery will depend on slower-growing species. Years, not months, for each recovered centimeter of skeleton. The timeline of coral recovery is a fact that's consistently underestimated. Massive corals, the round, compact species that are more likely to survive bleaching, grow between 1 and 2 centimeters per year. A colony the size of a human head may be a century old. A table coral, a flat, dish-shaped structure that serves as a platform habitat for dozens of species, can be 2 centuries old. When they die, they aren't replaced in years or decades. They're replaced over human generations, if conditions allow. With bleaching events now occurring every 2 or 3 years, the window for meaningful recovery is closing. Recovery can take years, and scientists warn that the time between events is shrinking, giving corals less and less time to bounce back. The reef is not just abstract biodiversity, it's biological infrastructure. The physical structure of dead corals persists for decades as calcium carbonate skeleton, still providing habitat and surface for new larvae to settle on. But without living coral for maintenance, that structure degrades mechanically from wave action and biological erosion. Boring organisms that inhabit the limestone speed up its fragmentation. The structural collapse of the reef eliminates the three-dimensional habitat that supports organisms unable to survive on a flat surface. The record losses in hard coral cover build on a string of other records in recent years, and mass bleaching events are becoming both more intense and more frequent, something observed simultaneously in 2024 and 2025. The reef that exists today is no longer the same reef that existed in 1980. Mosquito Bay, on the island of Vieques in Puerto Rico, holds the Guinness World Record for the brightest bioluminescent bay in the world, a title it has held since 2006. It contains up to 700,000 dinoflagellates per gallon of water, more than any other known bay on the planet. The organism responsible is Pyridinium bahamense, whose name in Greek literally means whirling fire. When these organisms are disturbed by movement, they emit a brief flash of blue-green light. The mechanism is purely biochemical. A molecule of luciferin reacts with oxygen in the presence of the enzyme luciferase, releasing energy in the form of photons. The bay maintains this concentration through a combination of physical factors. A narrow opening that prevents the organisms from dispersing into the open ocean, surrounding mangrove forests that supply nutrients, stable water temperature, and the absence of light pollution. Hurricane Maria caused widespread destruction on the island and effectively shut the bay down. To everyone's surprise, it didn't just recover, it came back brighter than it had ever been. To understand what makes Mosquito Bay so unique, you have to start with the organism that lives in it. The species Pyridinium bahamense, whose name translates as whirling fire, is a single-celled dinoflagellate that emits blue-green light when disturbed. At a density of 700,000 to over 2 million organisms per gallon of water, the combined effect turns the bay into a surface that responds to every paddle stroke, every touch, every fish that swims through it. Individually, each cell produces only 6 to 8 photons per flash, and the flash lasts about 1/10 of a second. The spectacle comes from sheer multiplication. Billions of simultaneous flashes turn the darkness into a glowing blue display. The biochemical mechanism behind this phenomenon operates inside specialized organelles called scintillons. Bioluminescence occurs when light-emitting compounds called luciferins react with oxygen molecules. This chemical reaction can't take place without the presence of an enzyme called luciferase, which acts as a catalyst. During the reaction, luciferin is oxidized, losing 2 electrons to become a compound called oxyluciferin. When oxyluciferin decays back into luciferin, it releases a photon of light. This light is referred to as cold light. It generates no heat, converting chemical energy into light radiation at nearly 100% efficiency, far beyond anything human technology has achieved. The biological purpose of this emission isn't to light up the environment, it's defense. When a predator swims close, the mechanical disturbance in the water triggers flashes around its body, making it visible to even larger predators. The dinoflagellate turns the act of being hunted into a signal that calls in an even more dangerous hunter. Ecosystems of bioluminescent dinoflagellates are rare, forming primarily in warm water lagoons with narrow openings to the open sea. The dinoflagellates concentrate in these lagoons, and the narrow opening prevents them from escaping. The entire lagoon can glow at night. Mosquito Bay brings all of these conditions together in an ideal combination. The S-shaped opening of the bay, so narrow it acts as a biological filter, traps organisms carried in by currents, preventing them from streaming out into the Caribbean. The surrounding mangroves are the second critical element. As they decompose, their leaves and roots release organic compounds that serve as nutrients for the dinoflagellates, sustaining dense populations. The bay's shallow depth keeps the temperature stable, preventing thermal stratification that would disperse the population vertically. The absence of artificial light. Vieques was a restricted military area for decades, preserved the organism's circadian cycle, which drives luciferin production during the day and emission at night. In 2017, Hurricane Maria devastated much of Vieques' infrastructure and deeply disrupted the bay's ecosystem. The disruption of the mangrove canopy altered the nutrient supply. Sediment churned up the water column. The bay went noticeably dark for months. The dinoflagellate population dropped during the end of 2017 after Maria, but began to recover and shot up to nearly 600,000 individuals per liter by June 2018, far surpassing pre-hurricane levels. The most widely accepted explanation is that the destruction of the mangroves, by releasing decades of accumulated organic matter from their roots, generated a massive pulse of nutrients that fertilized the population. The bay made a remarkable recovery after Hurricane Maria in 2017 and today shines brighter than ever. Mosquito Bay holds the Guinness World Record as the brightest bioluminescent bay in the world, a title it has held since 2006, with concentrations of 700,000 to over 2 million dinoflagellates per gallon of water. Swimming in the bay is prohibited to protect the fragile ecosystem. Visits are done by kayak with a transparent bottom or by electric boat, which minimize mechanical disturbance and chemical contamination. The swimming ban is a direct conservation measure. Oils from human skin and insect repellents alter the water chemistry and reduce population density. The bay's glow is not constant. It varies with the phases of the moon, seasons, and rainfall. On nights with a new moon, with no competition from artificial or lunar light, the contrast is at its peak and every movement leaves a bioluminescent trail for several seconds. On nights with a full moon, the water's glow loses visual impact even at the same organism density. Heavy rains temporarily dilute the bay, lowering salinity and disrupting populations. The organism's circadian cycle regulates when luciferin is available for emission. Production occurs during the day, even without incident light, controlled by an internal biological clock. Puerto Rico is home to 3 of the 5 documented bioluminescent bays on the entire planet. In addition to Mosquito Bay, there is Laguna Grande on the far northeastern end of the main island and La Parguera on the southwestern coast. These tiny bioluminescent organisms, fed by nutrients from the surrounding mangroves, glow when the water is agitated due to a chemical reaction. Dinoflagellates need a body of water that is protected from sudden swings in temperature, salinity, or turbidity in order to establish dense colonies. The fact that 3 of the 5 known bays are on a single Caribbean island is no coincidence. It's a convergence of coastal geology, stable tropical temperatures, and an unusually abundant mangrove cover in the region. The extraordinary concentration in Mosquito Bay defies any attempt at illustration by scale. A gallon of tap water weighs roughly 3.8 kilos. In that same gallon drawn from Mosquito Bay under optimal conditions, you'd find 700,000 living organisms, each one photosynthesizing during the day and emitting light at night. A microscopic civilization invisible to the naked eye, yet capable of making a person swim through a trail of blue light when they disturb the water. The concentration is the equivalent of fitting the entire population of Sao Paulo into a bucket-sized container, with every single inhabitant responding individually to every touch. The bay represents a rare example of an ecosystem that depends directly on its own fragility to exist. If the narrow opening were widened by any human intervention, the organisms would disperse into the open ocean. If the mangroves were removed, the nutrient supply would stop. If artificial light pollution increased, the visual contrast would disappear. If motorized boats replaced kayaks, fuel and oil would alter the water column's chemistry. Each thread is subtle on its own. In combination, any one of them could permanently extinguish the spectacle. Hurricane Maria demonstrated both the vulnerability and the resilience of the system, and it did so in a way that no theoretical model had predicted. Beneath the Antarctic and Arctic ice, on the underside of floating ice sheets, lives a community of microscopic algae, ice algae, that sustains the entire polar food web. Sea ice algae rapidly build up biomass, often at the base of the ice, growing into algal mats that are grazed on by amphipods like krill and copepods. Young krill congregate under the ice throughout the winter, and the algae provide critical food for them when there isn't enough light to produce food in open water. Rather than being a hardship, the winter ice is what allows krill to survive until spring. At the start of spring, the ice melts and releases the trapped algae, which then bloom explosively in open water. Scientists have estimated that the krill in the ocean surrounding Antarctica weigh more than the entire human population of the world. When the ice disappears, whether through accelerated seasonal melting or permanent loss due to warming, the roof of the subglacial forest disappears with it, and along with it the base of the entire food chain that sustains penguins, seals, and whales. Polar sea ice is not just frozen water. It's a three-dimensional habitat with complex internal architecture, brine channels, air pockets, rough surfaces, where complete biological communities live within a layer just centimeters thick. When the ocean freezes during the Antarctic autumn, microscopic algae are incorporated into the ice's crystalline structure as it forms. These organisms, mainly diatoms, which are algae with silica skeletons, become trapped but alive, adapted to survive at temperatures well below freezing and at the extreme salinities found in the concentrated brine channels that run through the ice. Young krill congregate under the ice throughout the winter, and the algae provide critical food for them when there isn't enough light to produce food in open water. Rather than being a hardship, the winter ice is what allows krill to survive until spring. This relationship is more precise than it might seem. Ice algae are nutritionally distinct from open water phytoplankton. They are rich in long-chain omega-3 fatty acids, a type of polyunsaturated fat that virtually no other source in the polar food chain produces at such concentration. Antarctic krill larvae depend on ice algae for up to 88% of their carbon intake during winter, when no other source is available. Imagining algae growing during a polar winter seems contradictory. The Antarctic winter means near total darkness for months. But ice algae have developed photosystems that operate efficiently on minimal light, capturing stray photons even when light intensity is a tiny fraction of full sunlight. The underside of the ice acts as a substrate surface, the equivalent of soil in land-based ecosystems, where the algae attach and grow into orange and brown mats visible when the ice is flipped over. These mats are dense enough to stain the underside of the ice in rust-colored hues. In spring, the ice melts and releases the trapped algae into the water. The algae, now living freely as phytoplankton, find everything they need to grow: open water, plenty of nutrients, and intense sunlight. The result is a phytoplankton population explosion. Animals, especially krill, consume this abundant food supply and multiply to extraordinary levels. This seasonal pulse of productivity is one of the most intense biological events on the planet, compressed into just a few weeks, feeding food chains that sustain mammals weighing tens of tons. Scientists have estimated that the krill in the ocean surrounding Antarctica weigh more than the entire human population of the world. Rich in protein and fat, krill become food for a huge number of animals up the chain, directly or indirectly feeding penguins, seals, and whales. That fact reveals the magnitude of krill as an energy link. A single blue whale, the largest animal that has ever existed, consumes up to 4 tons of krill per day. Blue whale, humpback, and mink populations that roam the Southern Ocean are entirely dependent on this crustacean, which in turn depends on ice algae to survive the winter. The Southern Ocean plays an outsized role in global carbon storage, accounting for up to 40% of the ocean's absorption of carbon dioxide. While most of this happens through physical processes, about 10% comes from primary producers like algae. Ice algae are a fundamental part of that bigger picture. When krill consume the algae and then defecate at greater depths, they are transporting atmospheric carbon to the ocean floor, a process called the biological carbon pump. Each ton of krill is, in that sense, a biological carbon capture mechanism operating at a continental scale. Sea ice ecosystems start with single-celled species, including bacteria, protozoans, and microscopic phytoplankton like algae. They grow on the underside of sea ice, which serves as the substrate. Miniature forests of microscopic organisms create a feast for small animals, zooplankton like copepods and Antarctic krill, that graze beneath the ice. The structure is analogous to an inverted rainforest. The canopy sits on the underside of the ice, light penetrates from above down through the ice, and the herbivores graze on the canopy from below as they swim. The loss of sea ice breaks this system at multiple points simultaneously. Without the ice substrate, the winter algae have nothing to attach to. The population never forms before spring. Without the winter population, young krill have no food during the critical months of their development. Without enough krill, penguins that synchronize their breeding with the peak abundance of the crustacean find less food for their chicks. The loss of krill, combined with the shrinking sea ice that Adelie penguins use as a feeding platform in winter, is a major reason why those penguin populations on the northwestern Antarctic peninsula have crashed by more than 80% over the past 35 years. The subglacial polar ocean is not separate from the global ocean. It feeds it. The dense water formed when the sea freezes and expels salt sinks to the ocean floor, driving the global thermohaline circulation, the current system that distributes heat, oxygen, and nutrients across all the world's oceans. Without this production of cold, dense water at the poles, global circulation would slow down, altering climate patterns on every continent. Polar ice is therefore both a local habitat and a regulator of the planetary climate system, and the algae living on its underside are the starting point of that entire chain of consequences. Along mid-ocean ridges, where tectonic plates pull apart, superheated water at 400 degrees Celsius pours from chimneys of metallic sulfides, while the entire surrounding food web runs without a single photon of sunlight. The foundation is chemosynthesis. Bacteria convert hydrogen sulfide, a gas toxic to most organisms, into organic energy. More than 700 animal species are found exclusively at hydrothermal vents. The giant tube worm, *Riftia pachyptila*, grows up to 2 meters long with no mouth and no gut, sustained by internal bacteria. The Yeti crab farms chemosynthetic bacteria on its own arms as a food source. Vent mussels maintain a dual bacterial symbiosis, drawing on both hydrogen sulfide and methane as energy sources. The hydrothermal vent shrimp, which has no eyes, has photoreceptors on its back, possibly to detect the faint infrared radiation emitted by the vents, a vision system that runs not on sunlight, but on geothermal heat. In 1977, geologists studying the East Pacific Ocean ridges descended in a submersible and found something no biological theory had predicted: oases of dense life in the deepest, darkest part of the ocean, with no source of solar energy whatsoever. The hydrothermal chimneys were expelling black mineral smoke at 350 degrees Celsius, and surrounding them, meter-tall white tubes, crabs, mussels, and shrimp were thriving at densities comparable to tropical coral reefs. The paradigm that placed photosynthesis as the mandatory foundation of all complex life was revised in real time. At hydrothermal vents, life depends on chemosynthesis, a process in which organisms obtain energy by oxidizing inorganic compounds. Hydrogen sulfide, H2S, the gas responsible for the smell of rotten eggs, lethal to most animals, is here a raw material for energy production. Chemosynthetic bacteria oxidize this gas, capturing the released electrons to build complex organic molecules. These bacteria completely replace the role of plants in land-based ecosystems. They are the primary producers, the base of the entire food chain at the vent. The most successful vent animals have taken chemosynthesis to the extreme. They have incorporated chemosynthetic bacteria directly into their own bodies. The giant tube worm Riftia pachyptila is the most dramatic example. These animals have lost their mouth, gut, and anus entirely over the course of their evolutionary history. Instead, they have a specialized organ called a trophosome, which occupies much of their body cavity and houses billions of sulfur-oxidizing bacteria. The bright red plume that extends from the protective tube is the animal's gill, absorbing hydrogen sulfide, oxygen, and carbon dioxide from the surrounding water and transporting them via specialized hemoglobin to the internal bacteria. Tube worms can grow up to 3 meters in length and were among the first animals observed when the vents were discovered, precisely because of their size and the distinctive red plumes emerging from their chitin tubes. The growth rate of Riftia is extraordinary for deep-sea organisms. It grows more than 85 centimeters per year under favorable conditions, making it one of the fastest-growing invertebrate animals known. This speed is possible because it doesn't spend energy hunting or digesting. Its internal bacterial farm produces nutrients continuously. The Yeti crab farms chemosynthetic bacteria in the hairs on its claws, called setae. In a remarkable example of agricultural symbiosis, it waves its claws near the vent openings to promote bacterial growth, then feeds on the microbes directly from its own claws. The rhythmic waving motion isn't random behavior, it's deliberate irrigation of the farm. By swinging its claws through the sulfide-rich hydrothermal fluid flow, the crab maintains the chemical gradient that feeds the bacteria. By pulling them back, it avoids lethal temperatures. The entire feeding strategy fits in the movement of a pair of hairy claws. The mussels Bathymodialis thermophilus are central to hydrothermal vent communities, forming vast beds that support and shelter a wide variety of organisms. Growing up to 20 centimeters long, these mussels have developed a unique dual symbiosis with 2 types of bacteria, one that oxidizes hydrogen sulfide and another that processes methane, providing them with multiple sources of chemical energy. This metabolic redundancy is strategic. Hydrothermal vents are unpredictable environments with variable chemical composition. Mussels with dual symbiosis can exploit whatever compound is available. The shrimp Rimicaris exoculata may be the most philosophically puzzling case of sensory adaptation at hydrothermal vents. Completely lacking external eyes, this shrimp has developed an entirely new light-sensitive organ mounted on top of its shell. The dorsal organ doesn't see, in any conventional sense. It is adapted to detect the blackbody radiation emitted by hydrothermal vents. For this shrimp, the dark ocean floor glows with the light of superheated hydrothermal fluid, allowing it to locate food for its own bacterial farm in its gill chambers and avoid getting cooked alive. Although essentially no daylight penetrates at 3,500 meters of depth, very faint light is emitted by the hydrothermal vents themselves. To make use of this light, Rimicaris exoculata has developed a modified compound eye on its dorsal surface that takes up about 0.5% of the animal's body volume. The radiation it detects is not visible to humans. It falls in the near-infrared range emitted by fluid at 350 degrees Celsius. This vision system works by detecting heat, not photosynthesis. completely flipping the logic of how organisms use light to navigate their environment. Hydrothermal vents also have a deeply unsettling temporal dimension. They are not permanent geological structures. They appear when a mineral chimney opens up, go dark when local tectonic activity stops, and reappear at other points along the ridge. Entire communities of organisms, including Riftia specimens decades old, can be wiped out locally within hours when a vent stops functioning. The larvae of vent organisms disperse through the water column in search of new active vents, a colonization process that resembles seeds searching for soil. The ability to colonize new vents on geological timescales defines which species persist in this ecosystem of ephemeral volcanic islands. The hydrothermal vent ecosystem matters far beyond its biological richness. The vents sit along ocean ridges, where tectonic plates are pulling apart and where magma superheats the water and dissolves minerals into it. These waters can reach around 400 degrees Celsius in the deepest zones, at roughly 3,000 meters of depth. The minerals that precipitate around the chimneys concentrate metals like copper, zinc, iron, gold, and silver at densities that surpass many land-based mines. At the same time, studying the chemosynthetic bacteria that thrive under toxic and extreme conditions provides models for the search for life on other planets, especially in subsurface oceans like the one on Europa, one of Jupiter's moons, where hydrothermal vents may exist at the bottom of an ocean that has never seen sunlight. The Sargasso Sea is built around floating sargassum algae that need no substrate to live and reproduce, and that structure a unique, free-drifting ecosystem in the open ocean that provides shelter and food for numerous endemic and vulnerable species. The sargassum supports 10 endemic species that have adapted to spend their entire lives in the open sea. It also acts as critical nursery habitat for many species of pelagic fish and every species of Atlantic sea turtle. But the most extraordinary role of the Sargasso Sea belongs to eels. It is the only known spawning ground for 2 endangered species of anguillid eel. Every single eel began life as a tiny leaf-shaped larva in the waters of the Sargasso Sea, before migrating thousands of kilometers across the Atlantic toward freshwater feeding grounds in the Caribbean, North America, Europe, and West Africa. Up to 25 years later, those same eels returned to the Sargasso Sea to spawn and die. No adult has ever been observed spawning there. Only the larvae found in the water confirm that it actually happens. The most basic question about eels is also the one that has gone without a definitive answer after more than a century of scientific investigation. No one has ever witnessed an adult eel reproduce. What is known was pieced together indirectly, with an almost detective-like elegance. In 1904, Danish biologist Johannes Schmidt conducted a series of expeditions in the Mediterranean and the Atlantic to investigate European eels. He discovered that the further west he sailed across the Atlantic, the smaller and younger the larvae he found. He sailed to the Sargasso Sea and captured the smallest eel larvae ever found. The conclusion was indirect but airtight. If the smallest larvae are there, the adults must spawn there. Eel larvae look nothing like eels. The fertilized eggs drift east on the Gulf Stream toward Europe, while American eels leave the Gulf Stream earlier to reach the east coast of North America, developing into transparent, willow leaf-shaped larvae called leptocephali. These larvae are flat, translucent, and so unlike an adult eel that for centuries they were classified as entirely separate fish species, with no one suspecting they were juvenile stages of the same animal. The metamorphosis that converts a leptocephalus into a glass eel, and then into a full-grown yellow eel, is one of the most radical physical transformations among all vertebrates. The Sargasso Sea plays an important role in the migration of catadromous species, such as the European eel, the American eel, and the American conger. The larvae hatch at sea and, as they grow, travel to Europe or the east coast of North America. Later, the the mature eel migrates back to the Sargasso Sea to spawn. The word catadromous describes exactly this pattern. Animals that live in freshwater but reproduce in the sea. The reverse of salmon, which live in the sea and swim upriver to spawn. The eel completes this entire cycle, from ocean larva to river-dwelling adult, and back to the ocean to die, without science yet fully understanding how it navigates. Researchers tagged 26 eels with satellite trackers from rivers in the Azores and followed them for periods between 40 and 366 days, providing the first direct evidence of adult European eels reaching their presumed spawning area in the Sargasso Sea. Before that study, published in 2022, the adult migration had been inferred. The larvae proved the eels got there, but no adult had ever been tracked to the final destination. The journey involves crossing the entire Atlantic at speeds of 3 to 12 kilometers per day, without eating. The silver eel, the migratory form of the adult, does not feed during the ocean crossing. The Sargasso Sea is geographically anomalous. Its defining quirk is that it is bounded by currents rather than continental margins. Formed within a massive ocean gyre in the North Atlantic, different water flows act as liquid walls, guiding and retaining whatever passes through the region and creating a sea with no shoreline. The four currents that bound it, the Gulf Stream to the west, the North Atlantic Current to the north, the Canary Current to the east, and the North Atlantic Equatorial Current to the south, rotate clockwise, creating a zone of relative calm at the center. That calm was described by historical sailors as a trap of winds and weeds. The Bermuda Triangle is part of this same region. Sargassum algae are not anchored to the seafloor, making them a mobile part of the ecosystem and habitat for a wide variety of organisms. Some species, like the sargassum fish and seahorses, have adapted to spend their entire lives in this floating environment. The algae also provide shelter for the young of many marine species. Loggerhead sea turtles frequently spend their first years of life in these floating mats. The sargassum fish, Histrio, is colored exactly like the algae, yellowish, with bumps and filaments that mimic plant textures. With no predator able to tell it apart from the sargassum, it spends its entire life inside the algae mats, eating anything smaller that passes through the same space. The European eel is classified as critically endangered. Its population has declined by more than 90% since the 1980s, due to a combination of river obstacles, dams that block migration, pollution, parasites, and ocean changes that affect larvae during their transatlantic drift. Scientists don't fully understand how these eels find their way to a precise location with no visible boundaries. The navigation mechanism remains an open hypothesis. Earth's magnetic field, temperature, and salinity gradients, or chemical scents from their home waters are all candidates. The eel emerges from a specific river in Europe, crosses the Atlantic for the first time with no prior experience, finds a sea with no shores, and descends, with no return, to die at depths where no adult has ever been observed in the act of reproduction. European and American eels begin their lives in the region, then migrate thousands of kilometers to rivers in the Americas and in Europe, a phenomenon that continues to baffle scientists. The separation of the two species from the same spawning ground is another mystery. The leptocephali of both species appear overlapping in the same Sargasso Sea waters at the start of their lives, with larvae of both species caught in the same nets. They then diverge. The American ones drift west with the Gulf Stream and exit earlier. The European ones continue along the North Atlantic Current and drift for 1 to 3 years before reaching Europe. Hybrids between the two species have been documented, confirming that their spawning areas overlap. The rarity of the Sargasso Sea as a defined ocean entity creates paradoxes in international governance. It belongs to no nation. It lies on the high seas, outside any exclusive economic zone. With no national jurisdiction, no government can regulate it directly. In 2014, 5 governments signed the Hamilton Declaration on the Sargasso Sea, committing to its conservation without having any legal authority over it. The plastic that accumulates in this zone of relative calm, carried by the same currents that bring eels, stays there for decades, entangling turtles and fish that no national law has the direct power to protect. Blue holes are limestone caves that formed when sea levels were lower during the ice ages, and were later flooded when the ocean rose. The most famous, the Great Blue Hole of Belize, is 300 meters across and 125 meters deep, with stalactites at 40 meters that prove the cave was exposed to open air 12,000 years ago. Below a certain depth, blue holes lose oxygen entirely. The dense, anoxic water at the bottom doesn't mix with the oxygenated water above. In that oxygen-free layer, decomposition stops. The extreme conditions inside blue holes preserve vital information about climate changes going back millennia, including ancient tsunamis, flash floods, and earthquakes. The anoxic sediment at the bottom accumulates stratigraphic records of climate events with a precision no aerobic environment can match. Each layer of sediment is a chemical snapshot of the year it was deposited. Studying this community offers a glimpse into the kind of conditions where life first arose on our planet, and may help guide the search for life in other water worlds in our solar system. To understand blue holes, you have to go back 20,000 years. During the last glacial maximum, enormous volumes of water were locked in the polar ice caps, and ocean levels were between 120 and 130 meters lower than they are today. What is now the Caribbean seafloor was at that time dry land. Limestone plains exposed to air, wind, and rain. Rainwater, slightly acidic from dissolving carbon dioxide out of the atmosphere, slowly dissolves limestone. Over thousands of years, that dissolution carved out complex underground cave systems, with stalactites growing from the ceiling and stalagmites rising from the floor, formed by dripping water carrying dissolved minerals. The Great Blue Hole was formed during several phases of quaternary glaciation, when sea levels were much lower. Analysis of stalactites found in the Great Blue Hole shows that formation occurred 153,000, 66,000, 60,000, and 15,000 years ago. These dates correspond to distinct glacial cycles. Each phase, when sea levels dropped far enough to expose the limestone, allowed a new round of stalactite growth. When the ice melted and the ocean rose, the cave was flooded, and the stalactites stopped growing. They remained suspended underwater, frozen in time, mineral witnesses to a different world. Descending into the Great Blue Hole, the first 15 meters are crystal clear, allowing a detailed view of the walls. Below that, the water darkens and the outlines of ancient rock formations appear, massive stalactites hanging like frozen curtains. Around 40 meters, sunlight disappears entirely, replaced by a quiet, still void. Divers encounter a layer of hydrogen sulfide, separating oxygenated water above from anoxic water below. That sulfide layer is a precise biological boundary. Above it, conventional oxygen-dependent life. Below it, conditions lethal to most organisms, but protective of the sediment record. The anoxic zone is the archive. In normal oxygenated environments, aerobic bacteria decompose organic matter that sinks, leaves dead organisms pollen, before it can permanently settle. In the anoxic zone, those bacteria can't survive. Any material that sinks below the sulfide layer stays intact, accumulating in sharp annual layers. Blue holes are excellent sediment traps and can preserve climate records and fossils dating back to the last glacial maximum. The reason they can preserve such records is the anoxic bottom water that most blue holes contain. Each sediment layer carries distinct chemical signatures. Hydrogen and oxygen isotope ratios that reveal air temperatures at the time of deposition, pollen species indicating what plants were growing in the region, and layers of disrupted coarse sand recording tsunamis or storms. In April 2025, scientists extracted a 30-meter sediment core from the Great Blue Hole, confirming the stability of its structure and chemical layers due to the anoxic bottom conditions that preserve historical records. That core potentially contains 10,000 years of uninterrupted climate history, preserved at annual resolution, something impossible in any aerobic land environment where disturbance, erosion, and decomposition erase the record. Hello there. Welcome back to Midnight Ocean. I'm Velen, your guide to traveling through the wonders of the ocean. Tonight, we're going to discover the bioluminescence of marine life in the ocean's depths. If these cozy moments calm you, I invite you to subscribe, share a comment, or leave a like. Now, get comfortable, take a deep breath. Don't be afraid, you're safe here. Let's dive deep into the sea and let our imagination guide us to a peaceful and deep night. Hello there. Welcome back to Midnight Ocean. I'm Velen, your guide to traveling through the wonders of the ocean. Tonight, we're going to discover the bioluminescence of marine life in the ocean's depths. If these cozy moments calm you, I invite you to subscribe, share a comment, or leave a like. Now, get comfortable, take a deep breath. Don't be afraid, you're safe here. Let's dive deep into the sea and let our imagination guide us to a peaceful and deep night. Hello there. Welcome back to Midnight Ocean. I'm Velen, your guide to traveling through the wonders of the ocean. Tonight, we're going to discover the bioluminescence of marine life in the ocean's depths. If these cozy moments calm you, I invite you to subscribe, share a comment, or leave a like. Now, get comfortable, take a deep breath. Don't be afraid, you're safe here. Let's dive deep into the sea and let our imagination guide us to a peaceful and deep night. Hello there. Welcome back to Midnight Ocean. I'm Velen, your guide to traveling through the wonders of the ocean. Tonight, we're going to discover the bioluminescence of marine life in the ocean's depths. If these cozy moments calm you, I invite you to subscribe, share a comment, or leave a like. Now, get comfortable, take a deep breath. Don't be afraid, you're safe here. Let's dive deep into the sea and let our imagination guide us to a peaceful and deep night. Hello there. Welcome back to Midnight Ocean. I'm Velen, your guide to traveling through the wonders of the ocean. Tonight, we're going to discover the bioluminescence of marine life in the ocean's depths. If these cozy moments calm you, I invite you to subscribe, share a comment, or leave a like. Now, get comfortable, take a deep breath. Don't be afraid, you're safe here. Let's dive deep into the sea and let our imagination guide us to a peaceful and deep night. Hello there. Welcome back to Midnight Ocean. I'm Velen, your guide to traveling through the wonders of the ocean. Tonight, we're going to discover the bioluminescence of marine life in the ocean's depths. If these cozy moments calm you, I invite you to subscribe, share a comment, or leave a like. Now, get comfortable, take a deep breath. Don't be afraid, you're safe here. Let's dive deep into the sea and let our imagination guide us to a peaceful and deep night. Hello there. Welcome back to Midnight Ocean. I'm Velen, your guide to traveling through the wonders of the ocean. Tonight, we're going to discover the bioluminescence of marine life in the ocean's depths. If these cozy moments calm you, I invite you to subscribe, share a comment, or leave a like. Now, get comfortable, take a deep breath. Don't be afraid, you're safe here. Let's dive deep into the sea and let our imagination guide us to a peaceful and deep night. Hello there. Welcome back to Midnight Ocean. I'm Velen, your guide to traveling through the wonders of the ocean. Tonight, we're going to discover the bioluminescence of marine life in the ocean's depths. If these cozy moments calm you, I invite you to subscribe, share a comment, or leave a like. Now, get comfortable, take a deep breath. Don't be afraid, you're safe here. Let's dive deep into the sea and let our imagination guide us to a peaceful and deep night. Hello there. Welcome back to Midnight Ocean. I'm Velen, your guide to traveling through the wonders of the ocean. Tonight, we're going to discover the bioluminescence of marine life in the ocean's depths. If these cozy moments calm you, I invite you to subscribe, share a comment, or leave a like. Now, get comfortable, take a deep breath. Don't be afraid, you're safe here. Let's dive deep into the sea and let our imagination guide us to a peaceful and deep night. Hello there. Welcome back to Midnight Ocean. I'm Velen, your guide to traveling through the wonders of the ocean. Tonight, we're going to discover the bioluminescence of marine life in the ocean's depths. If these cozy moments calm you, I invite you to subscribe, share a comment, or leave a like. Now, get comfortable, take a deep breath. Don't be afraid, you're safe here. Let's dive deep into the sea and let our imagination guide us to a peaceful and deep night. Hello there. Welcome back to Midnight Ocean. I'm Velen, your guide to traveling through the wonders of the ocean. Tonight, we're going to discover the bioluminescence of marine life in the ocean's depths. If these cozy moments calm you, I invite you to subscribe, share a comment, or leave a like. Now, get comfortable, take a deep breath. Don't be afraid, you're safe here. Let's dive deep into the sea and let our imagination guide us to a peaceful and deep night. Hello there. Welcome back to Midnight Ocean. I'm Velen, your guide to traveling through the wonders of the ocean. Tonight, we're going to discover the bioluminescence of marine life in the ocean's depths. If these cozy moments calm you, I invite you to subscribe, share a comment, or leave a like. Now, get comfortable, take a deep breath. Don't be afraid, you're safe here. Let's dive deep into the sea and let our imagination guide us to a peaceful. Analyzing that core is the equivalent of reading a climate diary for the region with annual entries covering the entirety of human civilization. The anoxic layers create habitat for specialized microorganisms, such as sulfidogenic bacteria or methanogenic archaea, that have adapted to survive in harsh, oxygen-depleted conditions. The research being conducted provides valuable information not just about adaptability, but has implications for astrobiology, suggesting how life might exist on other planets. Early Earth had an oxygen-poor atmosphere, and the first life forms were anaerobic. Blue holes are functional analogs of that primordial environment, preserved at the bottom of modern oceans as relics of ancestral geochemical conditions. Nematodes, predominantly non-selective detritivores, tolerate the anoxic conditions at the base of blue holes, allowing them to survive where other species cannot. They thrive in the deepest reaches of blue holes due to the abundance of organic matter that settles there. Similarly, foraminifera inhabit the lowest depths, and their diversity actually increases with depth. These microscopic organisms live at the edge of the sulfide zone, forming communities that make use of conditions that would kill any more complex animal. Studying them offers a window into the metabolism of organisms that lived on an Earth where oxygen was scarce, before cyanobacterial photosynthesis changed the composition of the atmosphere forever. Blue holes have also preserved a record of tsunamis with uncommon precision. When a tsunami wave sweeps through a coastal region, it drags sediment, coarse sand, and coral fragments into any nearby depression. Inside blue holes, these intrusive layers, chemically distinct from the normal slow-depositing sediment, are preserved between layers of fine sediment, marking the event with stratigraphic precision. Researchers have identified layers in some Caribbean blue holes that correspond to historically documented seismic events, validating the accuracy of the sediment archive. Deeper, more isolated blue holes that have never been disturbed by divers hold the most complete and pristine records available. The irony of exploring blue holes is that the act of studying them can destroy part of what makes them valuable. Divers disturb sediment by swimming near the bottom. Air bubbles locally alter the water column's chemistry. Artificial lights disrupt organisms adapted to total darkness. The Great Blue Hole is one of the best preserved natural records of sea level rise during the transition out of the last glacial maximum. Studies have found that the formation was flooded in several distinct phases. As the ocean rose, stalactites stopped growing and were covered by marine sediment, and corals began growing along the edges, marking each stage of the rising sea. Each layer is irreplaceable. Once disturbed, the chronology is lost forever. California's kelp forest is the only aquatic ecosystem where every vertical layer has its own distinct predators and keystone species. Macrocystis pyrifera grows up to 45 meters tall, forming a canopy so dense it filters sunlight into vertical shafts that cut through the water like rays in a Gothic cathedral. In the canopy, the sea otter floats on its back, wrapped in kelp fronds to keep from drifting while it sleeps. In the middle of the forest, sea lions and seals hunt fish. On the bottom, the leopard shark, Triachis semifasciata*, patrols the sediment. Sea otters are iconic keystone predators that can maintain kelp forests by preying on grazing invertebrates like sea urchins. Their arrival rapidly led to the depletion of urchins and the recovery of the kelp. Without otters, urchins eat through the holdfasts of the algae, and the entire forest collapses within weeks. Each layer, canopy, midzone, seafloor, holds a distinct community of species that depends on the structure created by the kelp to exist. Macrocystis pyrifera, giant kelp, is not a plant, despite every appearance of being one. It's a brown alga, more closely related to microscopic diatoms than to land-based trees. Its growth, however, rivals that of any known plant. In Monterey Bay, it typically grows 25 to 30 centimeters per day under favorable conditions. Under optimal conditions of cold temperatures and abundant nutrients, it can grow even faster. Each individual frond grows anchored to the rocky substrate by the holdfast, a root-like structure that doesn't absorb nutrients but locks the organism against currents. Nematocysts, gas-filled bladders, keep the fronds upright toward the surface, where light is greatest. The whole organism is a feat of solar capture engineering in a fluid environment. The three-dimensional structure created by the kelp is analogous to a mature forest on land, with distinct layers of light and shadow. The canopy at the surface receives full sunlight. Below it, a half-lit zone with filtered light. At the bottom, near total shade broken only by shafts of light coming through gaps in the canopy. The same Gothic cathedral-like columns that divers consistently describe. Each level of brightness selects for different species, forming vertically segregated communities within the same forest. Fish seek shelter among the kelp blades. Urchins feed on them, attracting in turn sea stars, otters, and other urchin-eating species. The feeding structure is simultaneously vertical and horizontal. Sea otters are considered a keystone species in the kelp forest. Their presence makes the entire ecosystem more resilient. Sea urchins can overgraze the kelp, especially when their populations spike. Otters eat urchins, keeping their numbers in check. Where otters are abundant, kelp forests thrive. Without otters, urchin populations can explode, devouring the kelp until nothing but bare rock is left. This kind of relationship, where a predator controls an herbivore that would otherwise destroy the entire habitat, is called a trophic cascade. The sea otter is the most well-documented example of a keystone species in marine ecosystems, studied since the 1960s in the Aleutian Islands. The image of a sea otter sleeping wrapped in kelp is iconic, but the biology behind it is functional, not decorative. The kelp forests along the Pacific coast of North America provide vital habitat for southern sea otters. An otter that let go of its kelp fronds while sleeping would drift out to open water or into coastal currents overnight. Wrapped in the algae, it stays in the same spot until it wakes up. That same frond serves as a cradle for pups while the mother dives to hunt. The otter leaves the pup floating on the surface, bundled in kelp, and descends up to 40 meters in search of urchins, crabs, and shellfish. Pioneering studies of sea otters and kelp forests in the Aleutian Islands showed that as otter populations recovered from near extinction, the otters turned urchin barrens into kelp forests as they recolonized the islands. The natural experiment of the Aleutians was revealing. On islands where otter populations re-established themselves, dense kelp forests replaced bare rocky bottoms dominated by urchins. On islands without otters, where historical hunting had wiped them out, urchins kept on dominating and no forest ever grew back. The presence or absence of a single predator completely determined the state of the ecosystem. The marine heat wave of 2014 to 2015, combined with a purple urchin population explosion, caused a dramatic decline of kelp in California, with roughly 95% of kelp forests in the northern part of the state disappearing. The collapse revealed the fragility of an ecosystem that had seemed robust. The heat wave warmed coastal waters, stressing the kelp and slowing its growth. At the same time, a viral disease wiped out sunflower sea stars, another natural predator of urchins, all along the west coast. With fewer predators and weakened kelp, urchins exploded in numbers and consumed what was left, turning forests into rock deserts. Kelp forest recovery requires specific conditions that are hard to recreate artificially. Kelp spores need clean rocky substrate, free of urchins and competing algae to germinate. With urchins dominating every available rocky surface, new spores simply can't find anywhere to settle. In Santa Monica, a restoration program combines science, daily dives, and direct population control of urchins to recover areas that had turned into bare rock fields. Kelp forests form a highly productive underwater woodland, serving as shelter, nursery, and feeding grounds for hundreds of marine species. Divers manually remove thousands of urchins per week, clearing space for the kelp to gradually recolonize. The kelp forest is also coastal infrastructure. The dense fronds absorb wave energy before it reaches the shoreline, reducing erosion and buffering storms. Kelp forests don't just play a fundamental role in holding back climate change. Sea otters and roughly 800 other marine species depend on them, as do fishermen in the state's abalone and sea urchin industries. Gray whales use the kelp forests as refuge from predatory orcas and as critical feeding grounds for their calves during migration from Baja California to Alaska. When the forest collapses, all of those functions collapse simultaneously. There is no substitute for the three-dimensional structure that kelp creates and that no other organism replicates. The sea otter and the sea urchin are the main characters, but the kelp forest is built by dozens of relationships that are just as complex. Garibaldi fish, vivid orange fish that hover in the midzones, aggressively defend territories, keeping shallow algae cleared where they feed. Moray eels coil in crevices along the bottom, waiting for unsuspecting fish. Bat rays sweep the bottom in formation, digging through the sand for shellfish. Octopuses shift color in milliseconds, confusing both prey and predators at the same time. Leopard sharks, harmless to humans, patrol the sediment in groups, eating buried invertebrates. Each species occupies a specific niche within the three-dimensional structure the kelp provides, and that vanishes entirely when the algae disappears. The coral triangle contains more than 76% of the world's shallow water coral species, 37% of reef fish species, 50% of razorfish species, 6 of the world's 7 sea turtle species, and the largest mangrove forest on Earth. A single reef in Raja Ampat, Indonesia, holds more coral species than the entire Caribbean Sea. The epicenter of this diversity is the Bird's Head Peninsula in Indonesian Papua, which alone is home to 574 coral species, 72% of the global total. Researchers believe the coral triangle may be the point of origin for many of Earth's coral species, the place where most modern corals first emerged before dispersing across the oceans. The relatively murky waters of the coral triangle appear to be protecting it from climate change, for now. The turbidity filters out some of the solar radiation that heats the water, creating a natural thermal buffer that has kept the triangle more resilient to bleaching than reefs in crystal-clear waters. To understand the coral triangle, start with a comparison that resets the scale of everything else. The Bird's Head Peninsula in Indonesian Papua is home to roughly 570 coral species, with individual reefs there, and in neighboring Halmahera, having up to 280 species per hectare. More than 4 times the total number of zooxanthellate scleractinian coral species found across the entire Atlantic Ocean. The entire Caribbean Sea, with all its reefs from the Bahamas to Venezuela, contains less coral variety per hectare than a single reef in Raja Ampat. This isn't a difference in degree, it's a difference in order of magnitude. The greatest coral diversity is found on the Bird's Head Peninsula in Indonesian Papua, which is home to 574 species, 95% of the coral triangle's total and 72% of the world's total. Within the Bird's Head Peninsula, Raja Ampat is the global epicenter of coral diversity, with 553 species. For reference, the entire Mediterranean is home to fewer than 100 reef-building coral species. Florida has around 70. Hawaii has roughly 60. Raja Ampat has 553, and that number has kept climbing with each scientific expedition that refines the local species inventory. There are 3 different theories about why the coral triangle has such species diversity, each proposing a different explanatory model. The center of origin model, the center of overlap model, and the center of accumulation model. The center of origin model holds that the high-diversity populations in the archipelago area are part of a centrally located ancestral population that later dispersed to peripheral locations. This model implies that the coral triangle didn't just concentrate diversity, it generated it. It would be the evolutionary laboratory from which most modern corals emerged before colonizing other oceans. Raja Ampat sits at the confluence of the Pacific and Indian Oceans, where powerful currents like the Indonesian through flow carry heat, nutrients, and larvae over great distances. Combined with stable ocean temperatures ranging from 27 degrees Celsius in the cooler months to 30 degrees Celsius in the warmest, these oceanographic conditions create an ideal environment for coral growth, fish recruitment, and evolutionary diversification. The Indonesian throughflow is a specific ocean current that transports up to 15 million cubic meters of water per second from the Pacific into the Indian Ocean. This massive flow carries fish and coral larvae, connecting populations separated by thousands of kilometers and continuously feeding genetic diversity. By analyzing coral bleaching, temperature, and turbidity data from 3,694 sites around the world using a Bayesian model, researchers found that turbidity within certain values reduced coral bleaching during thermal stress events. Roughly 12% of the world's reefs exist within this moderating turbidity range, and 30% of reefs with moderating turbidity are in the coral triangle. The mechanism is straightforward. Coral bleaching requires 2 simultaneous conditions: high temperature and intense sunlight. Turbidity reduces light before it reaches the polyps, breaking that lethal combination. Even in warm water, without full light, the photochemical stress that triggers zooxanthellae expulsion never peaks. Turbulent shallow reefs may help buffer some of these effects, as several studies have demonstrated their inherent resilience to thermal stress events. Future projections suggest that thermal stress events will likely become more frequent and severe over the next century, threatening the coral triangle's extraordinary biodiversity. Natural turbidity is not infinite protection. It's a buffer that buys time. In warming scenarios of 2 degrees Celsius above pre-industrial levels, the turbid reefs of the Coral Triangle will still be affected, just less frequently and with less intensity than comparable clear water reefs. Raja Ampat, 4 kings in Indonesian, referring to the 4 main islands of Waigeo, Salawati, Batanta, and Masul, is an archipelago of roughly 1,500 islands, islets, and reefs at the northwestern tip of the Bird's Head Peninsula in West Papua, Indonesia. The numbers are genuinely staggering. 553 coral species, 1,604 reef fish species, 700 mollusk species, and more mantis shrimp species than anywhere else in the world. The mantis shrimp deserves special mention. It has between 12 and 16 types of photoreceptors. Humans have 3. It sees color in the ultraviolet and near-infrared ranges, perceiving the visual world with a richness that no human instrument directly replicates. The threat to the Coral Triangle doesn't come from warming alone. Blast fishing and cyanide fishing, practices that are illegal but persistent, shatter coral structures built over centuries in a matter of minutes. The Coral Triangle has 15 regionally endemic coral species, found only there, and shares 41 regionally endemic species with Asia. These endemic species are irreplaceable. If they go locally extinct, they disappear from the entire planet. The concentration of endemism that makes Raja Ampat so extraordinary for science is exactly the property that makes every local extinction irreversible on a global scale. Conservation in the Coral Triangle faces the challenge that extreme biodiversity overlaps with human populations that are directly dependent on the reefs. More than 120 million people live within the 6 countries that make up the Coral Triangle, and a large portion of that population depends on reef fish as their primary source of protein. Raja Ampat is a living model of what marine conservation can achieve through collaborative governance, community engagement, and science-based policy. The network of 26 marine protected areas covering 22.5 million hectares was built with the active participation of local communities that depend on it economically. A different model from conservation that excludes human populations in favor of ecosystems. The Coral Triangle is also the world's largest sea turtle nursery. Of the 7 existing species, 6 nest on its beaches or use its reefs during some phase of their life cycle. Leatherback sea turtles, the largest, weighing over 600 kilograms, cross the entire Pacific and return to Papua to nest on the same beaches where they were born decades earlier, guided by Earth's magnetic field. Researchers named a newly discovered amphipod species found in the depths of the Mariana Trench Eurythenes plasticus, after the plastic found inside its digestive system at 7,000 meters of depth. Between the turtle crossing the Pacific and the amphipod digesting plastic in the abyss, the Coral Triangle connects the surface to the deep through chains of life that run the full length of the water column. The Great Barrier Reef lost decades of recovery in a single bleaching event in 2024. Bioluminescent bays exist because narrow inlets and mangrove forests created the exact conditions to concentrate organisms that light up at the touch. The subglacial polar ocean sustains whales with algae that grow on the underside of ice. Hydrothermal vents prove that life doesn't need the sun, just chemistry and heat. The Sargasso Sea is the graveyard and nursery of eels that travel 7,000 kilometers to die where they were born. Blue holes preserve ancient climates in oxygen-free sediment. California's kelp forest collapses without otters. The Coral Triangle concentrates 76% of all coral species in 1.6% of the ocean. And in the Hadal Zone, below 6,000 meters, where pressure is 600 times that of the surface, where plastic has been found at 11,000 meters of depth, amphipods and bacteria inhabit the deepest floor of the Earth with the same regularity that the sea otter floats in the canopy of the kelp forest, just a few meters below the sunlit surface. One continuous ocean, from the surface to the abyss. The ocean is not a collection of separate ecosystems. It is a single system with a continuous gradient of physical, chemical, and biological conditions. The otter floating in the canopy of the California kelp forest and the amphipod inhabiting the Mariana Trench breathe the same ocean water, connected by currents that take decades to complete their cycles. The carbon captured by Antarctic phytoplankton sinks, feeds abyssal amphipods, and some of that material eventually returns to the surface through upwelling. Nothing stays contained within a single ecosystem indefinitely. Scientific sampling has detected alarming levels of persistent organic pollutants in the tissues of amphipods collected at hadal depths, sometimes at higher concentrations than in coastal areas. These chemicals reach the deep ocean through a process called biological pumping, in which contaminated organic particles are consumed and transported downward through the food web. The plastic floating in Mosquito Bay affects bioluminescent bays. The plastic drifting through the Sargasso Sea reaches the hadal zone decades later. There is no boundary where contamination stops. In the Mariana Trench, lichenozoid amphipods collected at depths exceeding 10,000 meters contain synthetic microfibers and particles in their digestive tracts, with more than 70% of individuals from multiple trenches showing evidence of ingestion. These organisms have never seen sunlight. They live under 1,100 atmospheres of pressure, permanent temperatures of 1 to 2 degrees Celsius. They have never had direct contact with the surface, and yet they carry inside them the chemical signature of a century of human industrial production. Depth is not isolation, it is a destination. The way these 10 ecosystems work reveals a recurring pattern. Each one depends on a precise physical condition that, if minimally altered, undoes decades or centuries of biological structure. The bioluminescent bay needs intact mangroves and a narrow opening. The subglacial polar forest needs winter ice. The hydrothermal vent exists only as long as tectonic activity maintains the flow of hot fluid. Evidence suggests that turbulent coastal environments may offer some refuge through climate change, but those reefs will need high conservation status to remain viable near dense human populations. Each ecosystem is extraordinarily specific and therefore extraordinarily fragile. What connects the Great Barrier Reef to hydrothermal vents to the Sargasso Sea to the hadal zone is not just water. It is each one's dependence on conditions that exist because the planet operates within a narrow window of temperature, chemical composition, and geological activity. In 2020, researchers discovered a new amphipod species at nearly 7,000 meters of depth in the Mariana Trench. When they analyzed its stomach contents, they found a plastic microfiber and decided to name the organism Erythenes plasticus to raise awareness about the ubiquity of marine plastic pollution. Naming a new species after the plastic found inside it is a scientific gesture that documents, with taxonomic precision, the reach of human presence in the ocean. The hadal zone, the most extreme environment of all, harbors life because life found solutions to every problem the ocean floor imposes. The only known primary producers in the hadal zone are certain bacteria capable of metabolizing hydrogen and methane released by reactions between rocks and seawater, or hydrogen sulfide released from cold seeps. Otherwise, the first link in the hadal food web consists of heterotrophic organisms feeding on marine snow, both fine particles and the occasional carcass. The marine snow that feeds the hadal floor starts at the surface, where Antarctic phytoplankton fixes solar carbon. That same carbon that entered the ocean as sunlight arrives at the bottom as food for amphipods at 11,000 meters. A journey of decades through the entire water column. Each ecosystem described in this narrative is also a tool for understanding. The kelp forest demonstrates how a single predator maintains habitat structures that sustain hundreds of species. The blue hole demonstrates how anoxic environments preserve climate chronologies inaccessible in any other setting. The hydrothermal vent demonstrates that life can operate completely disconnected from solar energy. These specialized microorganisms from blue holes provide valuable information not just about adaptability, but have implications for astrobiology, suggesting how life might exist on other planets. Earth's ocean is the only available laboratory where conditions analogous to those of other water worlds, Europa and Enceladus Titan, can be studied with living organisms. The subglacial polar ocean and the hydrothermal vents represent the two extremes of the ocean's energy gradient. One depends entirely on sunlight filtered through ice. The other is completely independent of light. Between those extremes, the other 8 ecosystems occupy different positions on the spectrum of solar dependence, depth, pressure, and temperature. The Great Barrier Reef requires direct sunlight in shallow water. The Sargasso Sea draws on the productivity of the euphotic zone, but its primary role for eels plays out at intermediate depths. The Coral Triangle integrates shallow water, mesopelagic zones, and reef floors into a single vertical structure. Scientists studying the Antarctic marine ecosystem now know that its high productivity is concentrated at the edge of the sea ice and in a few other specific areas, rather than occurring uniformly across the Southern Ocean. Extreme productivity is not evenly distributed. It is concentrated at specific points determined by local physical conditions. The ocean covers 71% of Earth's surface, holds 97% of the planet's liquid water, and contains 94% of the habitable volume of the biosphere. Of that immense volume, only a fraction has been studied in enough detail to receive precise ecosystem classifications. Microplastics have been found in the digestive tracts of hadal amphipods in 6 of the deepest ocean trenches around the Pacific Ring of Fire, including the Mariana Trench, which contains the deepest known point on Earth. The reach of human contamination arrived before human understanding. We know more about the plastic that reached the bottom of the Marianas than we know about the species that live there. And new species are still being described with every expedition that descends into the abyss. The Great Barrier Reef losing coral to bleaching, the bay that lights up at the touch, the algae that sustain whales through the ice, the bacteria that live without sunlight, the eels that die where they were born, the caves that hold extinct climates, the forests that collapse without predators, the archipelago that concentrates life like no other point on the planet, and the abyss that receives plastic from surfaces it will never see. All are part of one single continuous ocean that circulates, connects, and transforms everything that enters it. The surface that reflects sunlight is the same water that, centuries later, moves through abyssal currents and resurfaces in another hemisphere as nutrients for phytoplankton that will never know the depth that fed it.